Yes.
Okay. Good morning, everybody. Thank you for that introduction. So, as was just said, my name is Hamza. I'm going to be leading today's talk on bioinformatics, a tale of the transcriptome. Before we even get started, all of the slides you're going to see today are shared through the Creative Commons license, which means that everything you see on every one of these slides can be adopted into your own presentations and freely distributed, provided that you give credit where credit is due. Now, in following with that, I'd like to give special thanks to Molokai and O.B. Griffith, who are principal investigators in New York and are fully focused on RNA analysis, as well as the Canadian Bioinformatics Workshop, which has helped develop a lot of these slides.
Hello there.
Can everybody mute their mics so that there's no echo coming back? Perfect. Now, just to get started, what is bioinformatics? Now, initially, I had a infinity gauntlet right on the left-hand side to try and give more of a description of what bioinformatics is, but each of the stones being a different infinity stone became this never-ending path on what's the time stone, what's the energy stone, so on and so forth. But the general idea is the same. Bioinformatics is the amalgamation of all of these fields coming together to solve hypotheses, especially in the advent of large data that's come from next-generation sequencing. While the field is absolutely massive, and there's hundreds of thousands of projects studying just different parts of the human species, let alone different species, we can establish a few common themes that bioinformatics can be broken into. These themes are genomics. Now, I will be using my laser pointer because I'm very visual in terms of a presenter and just as a person. So the breakdown of these different fields are genomics, which is the actual nucleic content in DNA, transcriptomics, which is the RNA that is then transcribed from the DNA, proteomics, which is the translated protein sequences from the RNA itself, metabolomics, which is the large-scale study of small molecules, commonly known as metabolites within cells, biofluids, tissues, or organisms, epigenetics, which is the control of gene expression through other mechanisms such as histone markers, methylation and acetylation groups, and pharmacogenomics, which is the study of pharmacological compounds on the entire system overall. You could spend a lifetime studying any one of these fields, especially because of how diverse they are and how diverse they are per species. But today's problems and hypotheses are often approached using more than one of these approaches to really gain a full perspective. So if you're looking at why a disease comes about, you may combine genomics and transcriptomics. You may combine transcriptomics and proteomics. You may combine genomics and epigenomics, so on and so forth. The combinations are quite large. However, for students, and what I've personally observed, the best or the easiest omics to approach, especially to get your hands on with bioinformatics, is RNA sequencing, so transcriptomics analysis. There are several reasons for this. The first of which is really simple. Getting access to transcriptomic sequences is far easier than genomic. Genomic sequences are very well protected, and you kind of need to work underneath a principal investigator who can then ask for those kinds of content. So it's just really easy to play on your own, and I'm a firm believer of you learn by doing. So it's best to just get out there and actually work with the data. But beyond that, why would we sequence the RNA instead of DNA, where everything starts? Well, for the first section, or for the first reason, it's really easy to do an RNA sequencing experiment when you're doing functional studies. Now, if you take a cell line and you treat it with a drug, the actual underlying DNA for both of these cell lines shouldn't differ by a lot. And the advantage of that becomes you don't want heterogeneity to play a larger factor, but you will still get a different phenotype. Maybe some cells grow faster than others. This isn't visible at the genomic level, but could be at the transcriptomic level. Alternatively, if you're studying a genome or you're studying a species that hasn't had billions of dollars invested into it, you might not actually have other investigators telling you what sections of that genome is transcribed and then eventually translated into proteins. So starting at the transcript level, you get more into figuring out what is actually going to result in actionable targets compared to the genome. Other times, just looking at the genome, you can't figure out what combinations of exons, which give rise to different isoforms, will actually be represented. Beyond that, fusion transcripts, which are transcripts that are separated by thousands, millions of bases, or even on completely different chromosomes, may come together to give you this novel transcript that is giving rise to your phenotype. Beyond that, it helps put mutations into perspective. When you look at something like cancer, which was my specialty before, well, still technically is my specialty, you can see mutations occurring throughout the genome. But for the most part, it's difficult to figure out whether these mutations even give rise to any kind of effect that is observable. What sequencing the RNA lets you do is if you know mutation occurs, say, further downstream in a region of the genome that may not really have anything interesting in it, you can see an increase or decrease in expression of a different gene further down. The reason for this is because of regulatory mechanisms that we have throughout the genome that may be affected because of that mutation. So it helps, this kind of goes with the idea of using more than one omics to figure out what and why a phenotype results from specific events. And then finally, you want to prioritize protein coding somatic mutations. So if you see a mutation, but the gene is not expressed, you're not really interested in that gene as much. If you see a mutation in a gene, but you only see the wild type, so the unmutated version of that isoform in the RNA, the reason might be that the transcript just has no function anymore. And again, it's less interesting, especially when you're trying to figure out why a phenotype arises, or if you're trying to find a target try and fix. If the mutant allele is expressed at the transcript level, this gives you a valuable target, especially with drug bindings in terms of cancer research. Although there's a lot of different kinds of approaches to RNA sequencing experiments, there are a few common analysis goals: gene expression and differential expression. In gene expression and differential expression, what you're basically doing, so this is an IGV track, just to visualize what it is. This first group here, think of this as sample A, think of this next one as sample B, and this is the reference genome that's been well studied, so we know that these are the transcripts. What you can do in RNA sequencing experiments is you can count how many of these transcripts you observed in sample A and sample B that line up with the exonic regions of this transcript, and then come up with a quantifiable number. Sample B has 10 reads, sample A has 50 reads. It helps establish just how much of a transcript you can actually observe. Because you can do this between samples, you can also ask questions like, if sample A and sample B had a different phenotype difference. Sample A grows faster than sample B. Is sample A expressed at a higher level than sample B with a statistical certainty? The latter part can't be more emphasized. Has to have stats involved. You can't just make claims that one is higher and one is lower. You can do this by using statistical models, but at the end of the day, you're just comparing the value of A versus the value of B. Alternatively, if it's a genome or if it's a transcript that hasn't been well studied and you align it to the reference genome, so each one of these small ticks is a read, you can actually figure out which exons are connecting with other exons and so on and so forth to figure out novel isoforms that you may not have seen before. Beyond that, you can do mutational discovery like we were talking about. In this case, these blue are the reads aligned from your experiment. And you can see that at this position over here, you actually have a T substituted for where a C might actually be, providing a novel mutation. And in this case, it's actually heterozygous, so one of your chromosomes has it. And as such, this might be an actionable target if it's the reason for why you observe a phenotypic difference. Lastly, you can use fusion detection. Now, this is a very concrete example. You have the EWS gene on chromosome 22, FLY1 gene on chromosome 11. Everyone has this, needed for normal function. What can happen, though, is you can have breakpoints happening in both of these genes where the resulting transcript is a combination of this first part of the EWS gene on chromosome 22 that combines with the second part of the FLY1 gene all the way on chromosome 11, giving you this novel transcript that isn't observed in nature. This transcript is actually responsible for Ewing sarcoma disease, and has been very well studied. Detecting this fusion transcript is almost always the cause of Ewing sarcoma and is almost an indicator. This helps emphasize that connections and so this is called a translocation, by the way. Translocations between transcripts can actually give functional effects after the fact. Although there are common analysis goals of RNA sequencing experiments, there are a few general themes of RNA-seq workflows. First and foremost, you need to obtain your raw data and convert your format into something that you can actually process on a computer. You need to align or assemble your reads. You need to process those alignments with a specific tool based on your hypothesis that you're asking. You post-process, so after you get quantifiable metrics for whatever question you're asking, you want to be able to run statistical tests to determine whether your values corroborate with the hypothesis in question. And then finally, you summarize and visualize because you need to be able to convey this information to someone else in a more distilled manner. Now, one of the most common kinds of RNA-seq analysis workflows is differential gene expression, where we try to determine what genes are differentially expressed between two groups. This goes back to knockout genes in mice or treating cell lines with a drug, where you want to see if the drug has caused certain genes to go up and certain genes to go down, and this might be shared in a common genetic pathway that is in the species of interest. The remainder of my talk will be focused on how you can actually go through these RNA-seq or the differential gene expression, but I'll be hitting each one of these points as we go through. A few of these examples of differential gene expression is you can go with, this is just to condense all the information as we go forward. Genome mapping, you take your reads, you map to the genome, then you can do transcript identification and counting, so you count how many of a read you've seen, or novel discovery. You don't know what the transcripts might be, or you might have discovered new ones, which is actually common in specific cancers. And functional annotation, you want to be able to attribute a specific pathway or pathways that may be going up or down that gives rise to your initial difference in groups. If your reads are really small, you can do transcriptome mapping, where you map just to the transcriptome rather than the genome. We'll get into this more as we go down in the further slides. And then reference-free assembly. So rather than aligning your reads, which is take all the small fragments of your reads and see where along the reference genome they map up, assembly is put all of your reads in order where you have overlaps between them. Try and string them together into larger consensuses so you get a contig. Then you take those contigs and you map them back to the reference genome. You count how many of each transcriptome you observe, and again, you do functional annotation. So before we even get to this last part, though, you need to be able to obtain your raw data. No matter what RNA sequencing experiment you actually end up running, you're never just going to naturally find data samples online that haven't come from a biological source. So you need to be able to convert your wet lab experiment into a format that can actually be converted into data, ones and zeros, or in our case, it's always going to be ATCG with a scoring matrix. I personally have never had experience doing these wet lab protocols myself, so I apologize in advance if something I say isn't 100% correct, but general protocols for RNA sequencing are available online, and you should always follow whatever the gold standards are, which are always documented in papers. But in general, for the most part, you take your tissues of interest, each tissue from the respective group, you isolate the RNA and you apply DNase, which tries to degrade all of the remaining DNA that is still inside, and you get this pool of RNA. It's a mixture of still genomic DNA that hadn't degraded, immature RNA, mature RNA, non-coding RNA, ribosomal RNA. Now, if you just take this entire piece and you put it into a test tube, you get total RNA extraction. Total RNA has broad transcript representation. You have abundant RNAs dominate, high unprocessed RNA, and high genomic DNA because you didn't filter for anything. And alternatively, you can do a ribosomal RNA reduction where you try and remove all these ribosomal RNAs. In this case, you still have broad transcript representation, but those abundant RNAs, the ribosomal RNAs, are de-emphasized. You still have high unprocessed RNA and high genomic DNA. Alternatively to that, you can do a poly-A selection where you're selecting for RNAs that have a poly-A tail. In this case, you have limited transcript representation because you're selecting for poly-As. You still have abundant RNAs de-emphasized because you're selecting against ribosomal RNA. You have low unprocessed RNA and low genomic DNA. Or alternatively, you do a cDNA capture, in which case you get a limited transcript representation, so it's targeted. Abundant RNAs are de-emphasized. You still have low unprocessed RNA, low genomic DNA. Which protocol you use becomes a question of what hypothesis question you're trying to answer, as well as what genes are you actually interested in. If you're studying ribosomal RNA, you're not going to do an rRNA reduction. That makes no sense. So the best practice is find papers that have done something similar and follow one of their protocols. If you go with total RNA, or if you go with any one of these, the general themes afterwards is as follows. You always need to assess the quality of your RNA. Running a simple gel and just observing your gel kind of tells you an overall perspective of how your RNA looks. These ones just giving you a few metrics. Intact mRNA, kind of a slew of different sizes. Total RNA, nice concrete bands, partially degraded. You kind of get these ladders, and highly degraded, slightly band, and then just all trickles to the bottom. Or a more quantifiable metric, you run an algorithm to determine your integrity. So it's called a RIN score, RNA integrity number. It's an algorithm for assessing integrity values to RNA measurements to try and tell you how well of a RNA extraction you did. I can't emphasize this more. With bioinformatics, it's garbage in, garbage out. If your wet lab protocol was subpar and you got kind of degraded RNA that you sent for sequencing, a lot of the conclusions that you're going to try and draw from that data might be inconclusive or might be wrong because you're providing inaccurate values to the further steps. Always, always, always make sure that every step can be maximized in terms of accuracy as you can. So going back, I just had to put that out there because it's almost intact. I've seen experiments that have taken years of troubleshooting because the analysis just didn't make sense, and it all traced back to, well, it was low quality data that they used. So always keep that in mind. Going back to this, once you have your total RNA, you can DNA treat to get rid of any DNA content that might still be there, and you enrich for specific segments. Over here on the right side, you can see your abundance of different sizes. So kind of like a gel electrophoresis of different RNA fragments that are present. You fragment the RNA, so you use sonication, try and break them up into specific chunks. So rather than have anything between 10 base pairs and 10,000 base pairs or 100,000 base pairs, you want to try and go after specific selection. This is important for sequencing. You want to uniform or a normally distributed size, somewhere between 300 and 500 base pairs, although again, it ranges based off of your hypothesis in question. After this, you can do a cDNA fragmentation, and you do size exclusion. And in this case, again, the height of the 35 base pairs and the 10,000 base pairs that you can see here actually got reduced because you're selecting, again, for this 300 base pairs, which is your RNA size of interest. From here, you have what you want to actually quantify. You add sequencing adapters, which are adapters that the next step is going to use, and then you send it for sequencing. Now, thanks to the advent of new technology, we have sequencers, these next generation sequencers from Illumina, from Oxford Nanopore, from Pacific Biosciences that are able to actually read these RNA molecules at hundreds of thousands, if not millions of reads per hour. And it has accelerated genomic research by magnitudes. Like it's honestly a revolution in terms of studying genomic sequences. It is quite complex on how they actually run and how they actually give you those read counts for a different lecture. All you need to know is that at this point, you send it to a sequencing facility, and they're going to tell you, or the machine is going to tell you what bases are represented at each one of these reads. A, T, C, G, C, C, A, T, and it also gives you a confidence score. That data you get back is what you need for your next step, which is aligning the reads to a reference genome or assembling them. To give you a more visual component, so these little blue marks are the reads that you get back. These are the fragments that you broke up and that the machine has read. In de novo assembly, what you're doing is you're lining up these reads in terms of what segments overlap. So this segment here and this segment here overlap between read 1 and 2. This small partial segment overlaps between 2, 3, and 4. And you do this until you have no more reads that kind of give you an overlap. This resulting sequence, if you read from the top below, gives you your overall contig. And this contig is what you then try and quantify between two different samples if a reference genome does not exist, or you can use these contigs to then try and figure out whether it maps to anywhere on the reference genome and see all the different mutations. Advantage or reasons why you use de novo assembly is, again, the reference genome does not exist for the species being studied. If complex polymorphisms, mutations, and haplotypes might be missed by comparing it directly to the reference genome. If you just look at these reads compared to the reference genome, it might align to a different place than it should, or it might be ambiguous and not aligned at all. Aligners have a lot of scoring metrics that they use to try and figure out what the best place is to line up with. If you have very short reads, what you want to do is you take your reads and you align them directly to the transcriptome for the genome of interest that you're looking at. So the caveat becomes you need to be studying a species that has a well-researched transcriptome. If you have longer reads or you have paired end reads, both of which I would highly recommend, what you can do is you can align to the reference genome directly, and then using the agreed upon transcripts for that reference, you can then quantify or determine whether you have transcript abundance, you have novel isoforms, because what you can literally do is where these reads map tells you, okay, read A maps, or read 1 here and read 2 here connected. So you know that the original transcript must have come here, jumped across, and then continued going here. So this is all one isoform. It allows you to ask more in-depth questions about your data, and the cost difference between short reads and paired end reads isn't that much compared to the gain in information you get. Kind of going on that idea as well to explain, sorry, I'm just going to jump back to explain this split over here and why this results. This is from the aligner's perspective. The aligner algorithm you see on the left side is STAR, which is a very well-known aligner. If this is the read that you get from the sequencer, what STAR does is it takes a chunk of the first read, sees where along the genome it actually maps. It finds an exact match and anchors it there. It then goes one base at a time, trying to build a continual match until it gets to a point where it can't map a larger segment anymore. At this point, when it tries aligning further, one base at a time, none of these bases match. So what it does is the last known correct match, it simulates a break in the read and starts a new read altogether on the right side. What this allows it to do is it takes a chunk of this read as if it was a completely separate one and finds where along the reference genome that read exists. Finds this anchor here and then continues building on it until the whole read is captured. These kind of aligners are known as splice-aware aligners. And the advantage is they know that exons can be separated by a large amount of genomic content because introns are spliced out. This also allows it to map confidently and then also account for mismatches that might happen, or anchor properly and then detect poly A tails, adapters, or poor quality ND. And emphasizing this point, so the read that you get from the sequencer is this mature RNA on the bottom here. But this mature mRNA actually has to look with the reference genome as this all the way on the top over here, exons separated by large amounts. So these two reads that were separated by whatever your target length, what shear length was, can actually be separated by a much larger distance. Splice-aware aligners know that introns are possible between exons. They require more memory. This exon over here would have no trouble either way. If the exon is large enough, your read maps confidently to both regions. There's no problems at all. Questions that you get asked, especially when you're setting this up. How much library depth? So how much do you have to sequence a specific sample for your RNA-seq experiments? It depends on a lot of factors. Are you looking at gene expression? Are you looking at alternative expression? Are you doing mutation calling? Beyond that, how well expressed has your gene been in other studies? What tissue type are you studying? Because that affects gene expression. What RNA preparation are you using? The quality of your input RNA. Again, if it's low quality, you're not going to see much. What library construction method did you use? From sequencing type, are you doing short reads, large reads? Are you doing paired or unpaired? And finally, based on all those factors, well, what kind of computational approach are you doing? And how strong of a computer do you have, or computational cluster do you have to be able to answer your goal? Some of these computational tools require a lot of memory and a lot of CPU power to be able to answer any of these questions. And if you just don't have those resources, you can't use those tools. The best rule is always identify publications with similar goals that have done something similar to what you're trying to achieve. The reason for this is these are peer-reviewed studies, and they're accepted in the general community as being a good practice for answering that question. Use those studies to help guide what approach you do for gathering your data, converting them, as well as sending for alignment. The general rule, though, for differential gene expression is about 30 to 40 million reads is common recommendation for determining moderate gene expression between samples. If your funding was large, or if you have money to spend, you should favor having more replicates, so having more samples, individual distinct samples that you're sequencing, rather than the same samples sequenced at a much deeper length. Now, this is more of a niche question, but it is applicable because the reads start off at a fragment and you're looking across the whole genome to see where it aligns. They can map to more than one location. The question becomes, should you allow the aligner to map it to more than one location? And that depends on your question. If you're doing variant calling, no, do not allow multi-map reads, because it might map to the original location that you were confident with, but has a mutation, and that's the one you should favor. If you're doing expression analysis, or if you're doing gene fusion discovery, the common accepted standard is to allow multi-map reads. For gene fusion especially, a read can actually, or a transcript can actually map to more than one location because there are multiple gene fusions. So you want multi-map to be on to be able to capture all of those. Once you have your reads aligned to the reference genome, you can then actually try and quantify them for whatever goal you want. You can use cufflinks for expression analysis. STAR actually runs an internal quantification, which we'll see in a second. Or you can do fusion detection. So trying to detect the EWS FLY1 mutation from before, you'd use a separate tool to figure out, oh, this comes from two separate genes, and I can assign a statistical certainty that, yes, this fusion is actually present. The way quantification works is as follows. These reads up here are what you get back from the sequencer and when you align them. So this reference genome you have below here is the well-known studied transcript, and you can see, oh, okay, gene 1 over here, exon 1, 2, and 3 has 1, 2, 3, 4, 5. 5 reads. You can assign a quantifiable metric in your given pool for how many mRNA sequences you may have had in your initial library. When you assign these values, 5 and, I don't know, like 25, 30, I'm actually not sure, it's quite large, to gene B, you can then do comparisons. But there's a few factors you have to take into account. The first is how long is the gene you're actually studying. If you have longer genes, let's say this wasn't to scale, if you have longer genes, they will naturally take up more room for more reads. And so the values will be higher compared to, say, a smaller gene. This doesn't take into account high or low expression. It just means that there's just more reads that can map to that specific region. So that has to be taken into account. Alternatively, if you send something for 30 million reads versus 60 million reads, the deeper sequence library will naturally have more reads per gene because you're just doing more cycles and trying to read it to a greater depth. If that was the case, and say you were comparing sample A down here and sample B up here, your conclusion would be sample A is highly expressed for every single gene compared to sample B, but that's because you haven't taken into account your depth of sequencing. So that is also a critical factor to take into account. Lastly, and this is a bit more complex and difficult to account, the read lengths you're using might make it difficult to map to specific regions, and repetitive elements in the region might make it difficult just naturally to align to that specific transcript. So it's not that the transcript isn't there, it's that the aligner has difficulty trying to figure out where it goes back to. Now, because all of these different factors are present, we need to do normalization. Again, why normalization? Well, as there are varying gene lengths in a species naturally capturing more reads and deeper sequencing libraries have more total read counts, normalization is required when comparing genes within a sample and between samples. This is where these typical acronyms you see for gene counts come about. A few of the most common ones are RPKM and FPKM, which stand for fragments, which are reads. It's interchangeable per kilobase of genomic content, so transcript, per million reads. The advantage of this is it takes into account comparisons between genes within a sample. Transcripts per million takes into account sequencing depth and gene length. You can do gene count comparisons within a sample and between samples of the same sample group. However, both of these, because you've taken them into account, you can't do differential gene expression analysis for them. Because you've normalized the values, you've kind of changed them. They're good for clustering, though. Lastly, DSeq2, which is a very well-known statistical package, has its own normalization, which is the median of ratios. The counts are divided by sample-specific size factors it estimates, determined by median ratio of gene counts relative to geometric mean per gene. What this allows it to do is you can do gene count comparisons between samples, and it's built for differential expression analysis. However, you can't do intra-sample comparisons. You can't compare gene A and gene B within the same sample using this method. The math is pretty straightforward for each one of these FPKM and TPM. FPKM, again, so just trying to get fragments per kilobase per million reads. Take all the reads in your sample, divided by 1,000,000. You just got your 1,000,000. Oh, well, divide the read counts by number 1. The steps are so quick that you can kind of do it in the same way. You divide your read counts by your number in 1, and you get fragments per 1,000,000. And then you divide it by the length of each gene in kilobases to get fragments per kilobase per 1,000,000. This you can use for clustering. Alternatively, for TPM, the order is kind of changed. You divide the read counts by the length of each gene in kilobases, so you get reads per kilobase. Count up all of the values in 1 and divide this number by 1,000,000. You get a per million scaling factor, and then you divide 1 by 2. The transcripts per million, the total numeric values, so the sum of all the values in a sample will always equal 1,000,000. So it's really easy to compare within a gene and between genes of the same group. So there's advantages to each one of these kinds of normalization strategies, but at this point, you have your matrix, you have your values, so you can do your post-processing. Import them into a different software package, R, MATLAB, Cytoscape, Ingenuity. Each one of these allowing you to run specific software packages to answer the question you initially had. In differential gene expression analysis, you can use DSeq2. If you're using Fusion or anything like that, you can do quantifiable statistics using one of these other packages. But because it's such a large aspect, or such a large unended question, it's literally the Wild West. You can use any number of tools to answer different kinds of questions, providing that your initial approach for library depth and for sequencing and your preparation are still sound. For differential gene expression analysis, you're tying gene expression back to a genotype or a phenotype, so tumor versus normal tissue. The question it's asking is what genes are expressed at a higher or lower level in different groups of samples with statistical certainty. The way DSeq2 does this is you have to provide it with the raw counts that came from your quantified aligned genome or transcriptome. You have to provide it with the sample phenotypes, and then you tell it what comparison you want to do. You can actually use multi-factor analysis. You can compare more than just treated and untreated. If there's a separate factor, if you treat it with a combination of two drugs, you can have 4 comparisons running at the same time, and DSeq2 will run a normalization within itself and then do a comparison between the different groups, assign a statistical certainty to that test, and then most importantly, it does p-value corrections. When you test tens or hundreds of thousands of times, you need to adjust your p-values due to multiple correction testing to figure out what truly is significant and what isn't significant. The paper's linked over there, and there's a lot of available modules to be able to go through and see this more in depth. Finally, once you actually have your data coming back, you can visualize and summarize your findings in either a cluster, a heat map cluster, or volcano plots. So let's say you did your comparisons between tumor or treated and untreated. The volcano plot here helps you visualize what's actually upregulated and downregulated. The x-axis here tells you your log2 fold change. So for every one value that goes up, you have a doubling in your experimental versus your reference group. At the same time, you can also determine which ones are and aren't significant from a p-value perspective over here. So negative log 10 of p-value, I believe the cutoff over here is 0.05. What this allows you to do is you can filter out the genes that are not significant, and you can also filter out the genes that don't have a large enough up or down regulation to have any interest from your hypothesis. The remaining genes are the genes that you then want to be able to take and determine gene lists or pathways or any other factors. Alternatively, if you made an FPKM or a TPM matrix, you can actually cluster them to see if you have any kind of phenotypic groupings still come about consistent. So in this case, normal looking surrounding epithelium compared to metastatic colorectal cancer and primary colorectal cancer. And you can see that this cluster of genes over here from these samples are actually more highly regulated compared to the cancerous parts, as well as this cluster of genes are downregulated compared to this cluster. And you can take these gene groupings to figure out whether pathways are going up or down. Now, this is basically a quick way of showing you how you can go through an RNA sequencing experiment. But I'm going to let my colleague Lauren run through a demo of what we just discussed using the BioBox platform, just to show you how quick and easy it can be. Although Lauren will be demonstrating transcriptomic analysis on the BioBox platform, it's currently capable of handling genomic analysis as well, all stemming from next-generation sequencing studies or even results. So in terms of getting your hands wet, I'd highly encourage you guys to use that. But without further ado, I am just going to switch this on. Give me 1 second.
BioBox is a software platform designed for anyone working with next-generation sequencing data. We provide an environment for data storage, complex bioinformatic pipeline, and data processing, as well as data visualization, so creating a wide variety of plots, data exploration, providing users with a place to find what is really meaningful in their data, as well as public data exploration. And the dashboard is the first page that users see when they hop onto the application. And this just includes a quick summary of all of the ongoing projects, the plots, which are dynamic and can be exported directly to your desktop, a summary of all of the people involved in your projects, as well as the ongoing progress. So one of the first things that you have to do when you're working with next-generation sequencing data is store and organize your data. And that can all conveniently be done from the BioBox library. When working with next-generation sequencing data, whether that be transcriptomic, so RNA sequencing data, or genomic, whole genome and whole exome sequencing data, one of the first hurdles that you have to overcome is how you're storing these large volumes of data, how it's being shared amongst members within your lab, as well as what your organization policies are for keeping track of things like cell lines and sample extracts that come from cell lines or patients. Within the BioBox library, we make this very simple. The first tab that we have here is the Models tab, and this is the overarching category that will connect all of your data points as well as your samples. For example, if you're a lab that works with patient samples, you can keep track of all of your patient data, anything from the age, treatment, date of birth to diagnosis. And let's say from those patient samples, you make cell lines. You can also keep track of all of your lab cell lines, including any metadata associated with them in regards to how they're grown, any supplements that are added, and what patient they are derived from. All of this information is fully customizable to suit the needs of your lab. Furthermore, if cell line and patient are things that you work with, you can create a custom model from scratch, which would fully adhere to your needs. In this example, let's say I need to run an RNA sequencing experiment, so I would take an RNA extraction from my cell lines. Those analytes would be held under the Samples tab. So samples would be any DNA, RNA, or protein extracts that come from my models that I would then send to the sequencing facility to generate my data. And you can keep track of all of the information associated with your samples just by hovering over the patient that is the source of the sample. So let's say I send these RNA samples to a sequencing facility for an RNA sequencing experiment. I can then upload my data to the BioBox platform. All data is stored on the Google Cloud platform, which is HIPAA compliant, so the data is safely and securely stored. Users have the option of uploading their raw sequencing data. So with respect to an RNA sequencing experiment, these would be FASTQ files. You can also upload BAM files. Or perhaps you processed your data elsewhere and now you have your processed data sets. These can also be uploaded to the BioBox platform, and you can quickly start visualizing as well as exploring these data sets. And currently we support differential expression, gene counts, as well as VCF and BAD files. And we are looking to expand all the types of processed data sets as well as sequencing data that we do accept. So once your sequencing data has been securely uploaded to the platform, you can start building complex pipelines in the cloud and getting towards visualizing as well as exploring your data. Our workspace is where you would go to create new projects where you can run bioinformatic pipelines in the cloud, as well as visualize and explore your data. We want to foster an environment for collaboration amongst lab members, as well as people in your organization. When you're creating a project, you have the option of having a project that's visible to all members in your lab or organization, a private project that's only visible to you, versus a limited project that's visible to a specific subset of people within your organization. Creating a project is very simple. All you have to do is give your project a name and select the visibility type, so we can create an organization-wide project and create the project. From here, you'll be directed to the project overview page. This includes the project title, any descriptions that you may give to it, as well as all members that are involved in this project specifically. On the next tab is our board, and this acts as a project board, so you can keep track of all ongoing progress within your project. You can assign tasks to lab members, as well as figure out what the status of a specific task is, including the priority. So once this has been set, you can simply drag your tasks into the associated columns to indicate what the progress is. So let's say you've uploaded your FASTQ data, and now you're ready to run an RNA sequencing pipeline. To do that, you would go to the Cube section. And the Cube section is where you would process all bioinformatic workflows as well as build and configure your pipelines in the cloud. To do so, you would create a new cube. And you'll be taken to the details page, which would include all of the metadata associated with your pipeline. And once you start to run your pipeline, all the input files that you're using to run the pipeline will populate here. And then on the Design tab, you have the option of either running a pipeline that someone in your lab has published to the organization. You could run one of the BioBox standard default pipelines, or you can create a new pipeline from scratch in our Pipeline Design Studio. This is our Pipeline Design Studio, and the goal of our Pipeline Design Studio is to alleviate a lot of the commonly faced pain points that scientists, biologists, as well as people who are new to working with next-generation sequencing experience. And if you're a bioinformatician, this often includes constantly installing new tools and updating your workflows to make sure that they are adherent to the newest and gold standards of the industry, and then running the pipeline, troubleshooting the pipeline via code, and then sharing the pipeline via bash script to biologists. Many biologists who aren't well versed in the world of coding often struggle to process this next generation sequencing themselves, and the collaboration between biologists and bioinformaticians can be greatly improved by having a common language and a common environment where people can seamlessly work and share things like pipelines as well as data. To build a new pipeline, the first thing that you would have to do is select your input files. In the example that we're going with, where we are building an RNA sequencing pipeline, our input files would be FASTQ files. So you would simply select the number of files you would like to use in this specific pipeline. If you click on the input, you can change the name and also provide a description. And perhaps this could be the workflow that you would like the specific file to flow into. If I'm running an RNA sequencing pipeline, the first step would be to use an aligner to align it to a reference transcriptome. Once I've added all of my input files, the next thing I would do is I would drag in the workflows that I would like this pipeline to run with. If I'm unsure of what any of these workflows do, I can simply hover over the workflow and I get a description of what this workflow does, what the typical use cases are, as well as what the inputs and outputs of the workflow will be. So in the event that I'm running an RNA sequencing pipeline and I would like to use STAR as my aligner, I would select STAR and it would be dropped into the center area. Here I would just drag and drop and connect the workflow input, so these FASTQ files will be flowing into the STAR workflow. And here I can see the outputs of the STAR workflow will be the sample gene counts file, as well as a BAM file. If I am well versed with bioinformatics, or I have experience running these workflows, I can additionally click on the workflow, and I can customize some of these advanced configurations. This would include the reference genome that my reads are being aligned to, as well as doing advanced things like adapter trimming or changing the file output types. The next step in building an RNA sequencing pipeline is to generate differential expression. So the gene counts will be used to determine what is changing between your two populations of interest, whether that be your control population versus your tumor population, or your treated versus your normal. So here I would select the differential expression workflow, and now I'm going to connect all the sample gene counts that are coming out of STAR and determine whether this is going to be the experimental or the reference group. So for the example where I'm submitting a set of patient samples for RNA sequencing, I would have a set of normal patient samples versus my tumor or disease patient samples. So here I'm just going to connect and confirm whether the samples are experimental or normal. And the output of this workflow will be a differential expression file that I can then use in things like scatter plots, bar plots, as well as exploring and figuring out which pathways are enriched or upregulated and downregulated, comparing my experimental and reference groups. And once I'm satisfied with the pipeline, I can save and exit. To run the pipeline, I select Launch. And now I'm required to input the files that I would like this specific pipeline to run with. And these are all files that the user has saved to the BioBox library. So from here, all I would have to do is drag and drop the specific data that I would like this pipeline to run with. Once I've dragged in all of the files I would like this pipeline to run with, I hit Submit, and the workflow will begin to run. On the progress page, in real time, I get an update of the status of all of the workflows in my pipeline, as well as any output files that are generated. Once the workflow is complete, I can head to the results page, and I can click on each workflow node, and I can see all of the successful stages that have been complete, as well as all of the output files. All of the output files are directly saved to the BioBox library, so the user does not have to worry about constantly downloading and saving their files. If they choose to, the data can be very easily exported directly to their desktop. So if you're running an RNA sequencing pipeline, one of the outputs of the pipeline is a differential expression file, and this file can then be used in scatter plots as well as volcano plots, and can be used to really explore and interpret and figure out what's going on with your data and what's meaningful from thousands and thousands of data points that you frequently receive from an RNA sequencing experiment. And to do that, you can visualize your data within a BioBox. So the BioBoxes are where we store all of our plots, as well as the key insights and very interesting biological findings that make the data meaningful. For example, one of the most frequently used plots when working with differential expression data is a volcano plot. So let's create one. When you create a BioBox, you're requested to select the type of visualization you would like to create. Right now, we currently support scatter plots, histograms, as well as heat maps. We're currently looking to expand our plot offering, and soon we'll be including things like lollipops, as well as Serco's plots. The next thing you would have to do is select the file that you would like to work with. So this could be a differential expression file that you uploaded to the platform, or a differential expression file that was generated as a result of running a pipeline on our platform. Once you select the data that you would like to work with, this page that you're taken to is the data table. And this contains all of the information found in the file that you selected. To create my volcano plot, I would go to the scatter plot, and I would specify what I would like to be on the X and the Y axis. And this relates directly to the data that you have in your data table. For a volcano plot, typically the log2 fold change is on the x-axis, and the p-adjusted can be on the y-axis. And here within seconds, you have your data, and when you hover over the data points, you get all of the metadata associated with those specific gene points. The plots are highly dynamic and can be fully customized. So if I wanted to add a title, for example, to my x-axis, I can quickly do that using the drop-down menu. I can also customize the graph to be any color that I would like. And I can also add a title. If I'm satisfied with my graph, I can quickly download the plot directly to my desktop, and it's ready to be inserted in a project, in a grant, or in a publication. So let's say I'm happy with the way my plot looks, but now I would like to look at genes that are specifically upregulated in the patients that I've sent for RNA sequencing. So I want to know what is changing in the patients that do have a disease versus that don't. To do that, I can filter my data. For example, I can use a log2 fold change greater than or equal to 2. And automatically my data is subsetted in the application. And if I go to my plot, now my plot has also changed to reflect this information. So now that I've filtered my data to only include all values that have a log2 fold change greater than 2, These are all of the data points that are upregulated in my patient tumor versus my patient normal samples that I sent for RNA sequencing. And now I'd like to know what specific pathways or what genes are enriched in this subset of data. And to learn that information, I can go to my Insights tab. One of the most valuable aspects of doing an RNA sequencing or next-generation sequencing experiment is to really pinpoint what pathways and what genes are changing in your experimental versus your reference samples. So this could be what pathways are changing with respect to the presence of a disease, or which pathways are changing in response to a specific treatment that you are giving to either patients or a cell line. And here we break all of that down under the Insights tab. We have 4 groups of pathways. Epigenetics refers to transcriptomic factors. Pathways are pulled from public databases K and Reactome. Ontology-related information provides insights from gene ontology, biological processes, as well as molecular function. And tissue and cells provides information from the Human Gene Atlas, as well as tissue expression, which is another GeoPublic data set. So now you no longer have to go through multiple databases and search engines to find what is meaningful and what is really valuable from your data. Furthermore, if you click on one of these pathways, you're taken to an in-depth look at that specific pathway. It tells you what genes are enriched in that pathway as well as in the data that you subsetted. Furthermore, it gives you any observations that you have on data that you either generated on the platform or uploaded to the platform. For example, I have a specific differential expression data set uploaded to the BioBox platform, and it's telling me that this specific gene is enriched and it's involved in this pathway. Furthermore, we give you an entire breakdown of the pathway, as well as a diagram explaining all of the pathway events. When working with transcriptomic or RNA sequencing data, a very common plot that's often made are heat maps. And heat maps can be very quickly and efficiently made within a BioBox. So once you've selected the data that you would like to work with, you can go to the heat map section of your BioBox. And the heat map is used as a visual representation of the expression levels of many genes, which correspond to the rows, across a set of samples, which are the columns. You can select how many genes you would like to fit into your specific heat map. And the heat maps are fully dynamic, meaning you can zoom in and zoom out to suit your needs. Additionally, you can adjust the cluster depth to be more or less stringent. You can also decide what method you would like to sort by, either by variance or by sum. And you can also customize the colors. And once you're satisfied with your heat map, you can export it directly to your desktop at any moment. But what's really valuable from the heat map is the information about the clusters of genes that are grouping together. And to get those insights, all you have to do is click on a specific cluster, and the insights for the specific cluster within your heat map that you selected will populate. Again, you can go through each one of these pathways in depth and click on them to get a better understanding of what is going on in the specific cluster on your heat map. So if I click on one of these pathways, it's telling me that these 3 genes were found in that cluster, and they're also involved in this pathway. I can look at my observations, and I can see that these are the observations in my heat map, as well as other data sets that I have processed on the BioBox. So by now, you've ran your bioinformatic pipelines, you visualized your data by creating scatter plots and heat maps, you looked at the insights, and you found what's meaningful in your data, and you found the pathways or genes that are driving your disease entity or that are being changed in response to a specific treatment. And now you're ready to learn a little bit more. Maybe this means comparing your data to third-party data sets or exploring public papers. At BioBox, we do have a place designated for doing just that, and it's called the Knowledge Engine. So the Knowledge Engine is a unified search engine for all of biology, and perhaps you're going through your insights and you found a really interesting pathway or you found a really interesting gene that caught your interest. All you have to do is search it in the Knowledge Engine, For example, if I found a specific gene called SOX10, and I'm studying the disease glioma, and here I'll be taken to my search results. On the overview page, you get a subset of some of the papers associated with your search, as well as a gene network interaction map involving your gene of interest. The Papers tab provides you with a comprehensive list of all the papers associated with your search, and at any moment you can direct to the PubMed link. The Gene tab provides you with all of the gene metadata, including the chromosome start and end site, description, and other information of that nature. However, what really differentiates us from other biological search engines is our Insights tab. Because the Insights tab is pulling information directly from all of the data that you have processed on the platform or that you have uploaded to the platform and is providing you with key observations. For example, the gene SOX9 is associated with my search, and within my observations, I have a data set that has a log2 fold change of about 1.9 on the specific gene. Same thing goes for MIC-N, and I can see all the observations, and if I do have multiple data sets with observations of these genes, all of the information will appear here. Additionally, you do get further information about associated pathways, as well as observations found in those pathways, as well as in your data sets. In summary, the goal of BioBox is to be an end-to-end solution for anyone who's working with next-generation sequencing data. This can be anything from genomic to transcriptomic data, as well as for users who are bioinformaticians and who are well-versed in running complex workflows, to scientists and biologists who are new to the next-generation sequencing space, as well as soon-to-be graduate students who are maybe just learning about how the entire landscape works. As an end-to-end solution, we want to provide a safe and secure environment for collaboration, data storage, running complex bioinformatic workflows in the cloud, data visualizations, so creating a wide variety of plots, a place for data exploration to really find what's meaningful in your data, as well as discover new things in public data. And this is just our initial feature offering. Over the next few months, we will continue to release new plot offerings, new workflows, as well as really cool and awesome features that we would love to get the scientific community's feedback on. Thank you. BioBox is a
So that concludes our presentation. And thank you to Lauren for giving us that demo. I encourage everybody to go out and get their hands wet with RNA sequencing data, especially to really solidify the topics we talked about in today's talk. There's a multitude of other lectures and resources you can use to really dive into it. This, in my opinion, this entire lecture can actually be broken into 2 days worth of talks and practice, just dealing with transcriptomics and not even touching genomics or any of the other fields. So the more practice you do, the better. So check out the Canadian Bioinformatics Workshop for the actual, for more presentations on how you can actually go about looking at data. And everybody who's attending today is able to sign up for the waitlist for BioBox. So you actually have a platform to go and apply anything you learn and really dig into the data that you may find either through the Canadian Bioinformatics Workshop or through public resources. Thank you to everybody for attending, and thank you to BioCord for inviting us to be able to give this talk. And feel free to send any questions you may have that we can answer for you. Thank you. Thank you.